A research video on heterotrophic nanoflagellate ecology by Gabrielle Corradino at North Carolina State University. The world's oceans cover 60% of the globe, yet more is known about the surface of the moon than certain parts of the deep sea. Within the ocean lies one of the foundational cycles that we depend on, the carbon cycle. Let's take a look at this system. The carbon cycle starts at the top layer of the ocean where there are high amounts of sunlight. The light energy and atmospheric carbon dioxide allows for photosynthetic organisms like phytoplankton to activate their pathways and produce food and oxygen. These organisms reproduce and populate the photic zone. As they die, the algal cells clump together and sink throughout the water column. These particles, along with other sinking material, become dissolved organic matter. These aggregates of dead cells attract heterotrophic bacteria, viruses, and nanoflagellates. The bacteria actively break down and consume the organic matter. These particles are microhabitats for these different bacterial species and for flagellates. Flagellates, which can range in size from 2 to 20 microns and often have multiple flagellum, act as a top-down control to bacteria. In addition to consuming bacteria, nanoflagellates can also attach and parasitize phytoplankton. My research examines the dynamic relationship between nanoflagellates and phytoplankton species. The video that you see here is an example of a specific species of heterotrophic nanoflagellates in a dying algal cell. There are over 30 anchored and attached flagellates that are actively consuming this algal species. These flagellates act as a primary link between the dissolved organic matter and the microbial loop. It allows for nutrient remineralization. This remineralization allows for these nutrients to be shunted up the food web from the microbial loop back into the carbon cycle. The interaction between phytoplankton and nanoflagellates is not rare. Between 90 to 92% of diatom species, which are a type of algae, actively have heterotrophic nanoflagellates attached to them. Understanding the trophic level interaction between these flagellates and the bloom-forming phytoplankton is pivotal in a time of global climate change. The IPCC stated that in the 21st century, the CO2 emissions will double. With an increase of sea surface temperatures and UV penetration, the microbial food web dynamics will be disrupted. In summary, my research will use multidimensional experimental approaches to answer questions on heterotrophic nanoflagellate behavior and the interactions with different phytoplankton species. This will begin to elucidate planktonic community dynamics and help us to truly understand how the flow rates of carbon may be altering within the microbial loop during a time of global climate change.